Thank you for joining our 5th Data Sharing and Data Reuse Seminar sponsored by my office, the Office of Data Science Strategy. Our goal and mission is to create a modernized and integrated, fair, findable, accessible, interoperable, and reusable biomedical data ecosystem. Thanks to all of my colleagues at NIH who work with us to create this vision. Our next slide is just a little bit about how you can interact with us during the seminar. We like to use Slido. You see the hashtag there, NIH Data Senpai, so that we can upvote, downvote questions. So it's really interactive. So if you do have a question, please do post it in Slido. If you can't find the link, there's a Q&A box here, but we do hope to use Slido. Next slide, if there's a next slide. Just a little plug. Our next data sharing and data review seminar is sponsored by the National Library of Medicine, and it is Dr. Mark Newson. He will talk about topics to integrate tools and to make biomedical data more fair. You can see all of our past seminars and their recordings just by going to this link here on the slide. So do join us on July 9th. The next slide. All right, so this is where I am going to introduce my colleague, Dr. Susanna Petchenka. She joined the National Institutes of Aging in 2005 as a program director in the Division of Neuroscience. During her tenure at NIA, she oversees and develops a number of programs in basic and translational research for Alzheimer's disease. She's been instrumental in the development of NIA's AD, Alzheimer's disease translational research program, the epigenomics of AD, and the acceleration of medicine partnerships for AD, the AMP program, particularly focused on target discovery and preclinical validation projects. Since 2012, Dr. Petchenka has led a number of NIA's strategic planning activities related to achieving the research goals of the National Plan to Address Alzheimer's, to prevent and treat AD by 2025. Dr. Petchenka is a graduate of the University of Belgrade in the former Yugoslavia, where she received her bachelor's degree in molecular biology and physiology. She holds a Ph.D. in pharmacology from New York University. Following her postdoctoral training at Rockefeller University and Cornell, she established her independent research career at the Nathan Klein Institute in Orangeburg, New York, and she joined the faculty of New York University Medical Center. Her research focuses on the role of disruptive steroid metabolism in Alzheimer's disease pathogenesis and on the mechanisms by which estrogens and cholesterol-lowering drugs might exert neuroprotections. Thank you so much, Susanna, for introducing our speaker today and for all the work you're doing to address Alzheimer's. So important. Thank you.
Thank you very much, Susan and Erin, for the invitation to contribute to this wonderful seminar series. So we can, what I thought I'd do in the next few minutes is actually share how our institute uses data science to accelerate therapy development for Alzheimer's. Next slide, please. We are the largest funding agency for Alzheimer's research, and one of the major strategic goals for the institute is to enable precision medicine approach to AD treatment and prevention. A formidable challenge given the syndromic nature of the disease, the fact that there are multiple etiologies of play, long prodromal period, and multiple progression trajectories. Next slide. So, of course, we're armed with, to execute on this lofty goal, we're guided by a whole series of strategic planning research summits that inform our program development activities, and we have benefited from a tremendous increase in public funding over the past 6 to 7 years. Next slide, please. So our therapy development programs in Alzheimer's research operates through two major components. So we support the development of new preclinical and clinical drug candidates through a pipeline of funding initiatives, and this is a portfolio that has been rapidly growing over the years. But equally enabling, and actually critically enabling, are a set of enabling infrastructure programs that are heavily marked with the presence of data science and actually utilize data science to enable all of the steps of translation across this critical path. Next slide, please. On the very front end, obviously, is our very robust genetics and genomics programs, and the flagship initiative here, the Alzheimer's Disease Sequencing Project. The overall goal here is to resolve the complex genetics of AD, but also to provide these key insights and foundational data and insights to enable a precision medicine approach to therapy development. Some of the newer ADSP-associated programs are heavily focused on data harmonization, functional genomics, AI, and the most recent one just launched an initiative that will take this program into a whole spectrum of diverse genetics of diverse populations. The data infrastructure supporting all of our genetics programs is NIGAS, the NIAID genetics storage site. Next slide, please. We also have an entire series of enabling infrastructure program in the translational space, spanning from target discovery to clinical trials that are at their core are built around open science and open source principles. Next slide, please. And at the very front end of this series of programs are our Open Science Systems Biology Consortia that started with the Accelerating Medicines Partnership for Alzheimer's Disease, and you'll hear some detail on this from Laura. And early taking, really harnessing the power of big data and open science principles to generate insights around new targets and biomarkers. And the central feature of these programs is the very rapid and broad sharing of data, methods, and results through a centralized data infrastructure, namely the AD Knowledge Portal and the portal-linked Agora open source platform. And again, you will hear from Laura in some detail on this. Next slide, please. So building the output of the investment in genetics and in the Open Science Systems Biology Consortia is really utilized, and it has enabled us to build a whole set of translational centers, the model AD centers that are developing the next generation animal models that are informed in human genetics and are aiming to model aspects of late onset AD, and the treat AD centers that you'll hear about from Laura that are trying to enable novel targets for drug discovery campaigns through developing, prioritizing targets and developing open source target enabling tools. Next slide, please. So just a couple of words on that, as you all know, open science and data sharing is not something that happens spontaneously. It's a very proactive, deliberate process, something that we have invested a lot of thought and effort at our institute and across the NIH, just to share some of the elements on components that have proven effective for us, starting from very clear and enforceable expectations vis-a-vis funding opportunities and terms and conditions of funding, really emphasizing no embargoes on data reuse, no IP barriers for sharing research tools. Also, the critical importance of building and investing a fair and sustaining a fair data infrastructure that can not only support the sharing of complex data types, but also a diverse set of end users. Critically important has been for us that to provide support for data sharing and data management activities, and last but not least, training and community building. Next slide, please. And so something that the benefits of actually enabling this open science research ecosystem is that we have been able to very rapidly develop some downstream programs on the output of the Open Science Consortia. Just here to show that in just short 3 years, we were able to build a very robust translational bioinformatics program focused on advancing drug repurposing and combination therapy for Alzheimer's disease. And although these projects are using the entire universe of available public data, they're heavily leaning on the data sets coming from AMP-AD and the associated consortia, and our genetics initiatives. Next slide, please. Training, so we're trying to bring data science into the training curriculum for the next generation of translational scientists. And here is one of our training initiatives. It's an institutional training grant where we're incentivizing institutions to build programs at the intersection of data science, basic and clinical research, and drug discovery. And on the next slide, also a pipeline approach to, or creating a pipeline of the next generation translational scientists. Here is a series of just released funding initiatives where the goal of which is to develop a diverse translational research workforce that can either participate or lead team science precision medicine research on AD. And here again, the emphasis is to develop skills in data science and drug discovery and apply these to various aspects of AD research. And finally, the next slide. So we continue to raise the bar on data sharing and continue to communicate this with our researchers. Here are just some of our blogs where we try to help the community keep abreast of what our expectations are and help them be prepared for a world where open science is just another name for good research. So I will end here, and it is my distinct pleasure to introduce Lara Mangravite, President at Sage Bionetworks. Lara is the principal investigator for the Accelerating Medicines Partnership for Alzheimer's Disease Data Coordinating Center, and also a leading investigator of one of our Treat AD centers. Lara is a real rocket scientist, starting with a Bachelor of Science degree in physics. So Lara, please take it away.
Thanks. I'm so pleased to be here. I appreciate the opportunity to talk with all of you about the work that we've been doing. All right, is that showing up in the right orientation? Actually can't.
Yes, that's correct.
Okay, great. So that was a perfect setup. So I appreciate that, Susanna. And so let me get started. All right. So one of the wonderful things in the field of Alzheimer's disease has been this increased investment, this embarrassment of riches that has allowed Susanna and the other program officers at the National Institute on Aging to really start to think at scale about how they want to address the problem of Alzheimer's disease and the problem of treatment. There are also a whole series of private funders who are really getting into funding of Alzheimer's disease research right now, or have been over the last couple of years. And so it's this amazing time in the field where we can start to think about taking on approaches and questions that we haven't been able to take on given resources previously. One thing that we've seen, not just in Alzheimer's disease, but actually across the whole research ecosystem, is that frequently in a time of increased investment, the tendency of the way we set up our scientific system is for people to do increased analysis on the things they're already working on versus expanding into other potential avenues, in this case, talking about understudied Alzheimer's disease mechanisms. And so we see this trend in our field as well. So what I'm showing you here is a graph that shows all of the genes that have been linked to Alzheimer's disease across the x-axis and the number of publications that are being published on the y-axis. And simply the point here is that we continue to study now the things that we studied 50 years ago, 20 years ago. And what we'd really like to do with the products and programs and services that I'm talking about today is to catalyze exploration of some of these understudied Alzheimer's disease mechanisms. So we'd like to encourage researchers to be moving into unexplored space. And we'd like to do that in two ways. One is we'd like to expand this set of genes, of course, that is linked to Alzheimer's disease. And then two, we'd like to shift the graph out into the part that currently is the long tail of exploration. So the opportunity is here. So how do we go about doing this? How do we catalyze the study of understudied mechanisms, pathways, targets, trying to advance treatment? Everybody on this call, I'm sure, has seen this week FDA approval of aducanumab, which is treating the amyloid hypothesis. This is obviously an amazing milestone in the field. And so this, I guess what I'm saying is not that we should stop studying mechanisms like that, but we know this is a very heterogeneous disease. We know it may in fact be a collection of diseases that are showing similar symptomologies. And so we need to be looking at a diverse set of targets and pathways as we think about how to understand, prevent, and treat the disease. And so what I'm going to talk to you about today are some of the open science approaches we've used to leverage data and tools to address this question. So this process really started as the brainchild of Susanna and her colleagues with the Accelerated Medicine Partnerships in Alzheimer's Disease about 7 years ago. And here, the NIH, the FNIH, and the NIA came together to create a private-public partnership that was designed to use cutting-edge systems biology approaches on human data to evaluate pathways and mechanisms and identify new Alzheimer's disease targets. This is to complement the wonderful work going on the genetics consortia and the sequencing projects within Alzheimer's disease. And so, as you can see here, the goals were to identify target predictions catalyzed through systems biology approaches, and then to do objective evaluation of these target predictions using open science. And the way the program was set up was to fund a series of projects. So you can see 6 of them listed here. The projects and teams have expanded and changed over the years. And each of these projects was set with the goal of generating multi-omic human data, largely from brain samples, although there are some other tissue types, to then use their own best practices in terms of systems biology or network analysis to characterize the molecular state of disease and to identify potential targets or pathways involved in progression, and then to develop their own systems to validate some of the computational hypotheses that they have come up with. And so I think this is a really interesting approach as you're moving into a new field where methods actually are not mature enough to be standardized, and each of these groups had their own approach. Rather than going and funding one big project with one group doing the work, to ask multiple groups to take their best shot on goal, and then to bring them together in one of these open science, in an open science environment to evaluate, compare, and collect evidence across all of the various groups and approaches that they were taking. So our role in this project was to serve as the data and the analytical coordinating center for the work that was going on. And so I wanted to emphasize each of these very talented teams is going after this same question with their own approaches, and they are each coming up with their own data, their own tools, their own results, and a whole robust set of publications and innovative research findings. And one of the things we did as this project went on was to ask them repeatedly and continuously to be sharing in a pre-competitive way the information that they were collecting. So not just the data and the tools, but the outcomes as well. And we were able to aggregate them quite easily for the whole community, the whole consortium to look at. And so what I'm showing you here is a graph coming out of our open results portal for this particular project, demonstrating the outcomes of these projects. So all of these various groups identified Alzheimer's disease targets, they deposited them into this collective set, and we looked to see what the overlap was in terms of what each of these groups were finding. And it was very interesting to look at in the very beginning. We asked each of the groups to identify, I think it was about 100 targets, and there was some overlap in what they found, but not a lot. And initially, this was concerning, particularly to some of the private funders who were saying, we're really looking for interesting mechanisms that we can follow up with, in our companies, and we're not sure what to do when the 6 teams are actually advancing different targets, and we can't, how are we supposed to pick if you're not all agreeing on the same thing. And so I bring that up because that wasn't actually, what I'm going to show you is that wasn't actually an issue with lack of replication. It was actually more of an issue of how we as scientists operate, right? So A, we're storytellers, and B, we're not all going to pick the same thing to go after. So, I'm going to tell you how we used open data and collaborative meta-analysis across these groups to establish how each of these groups was in fact either supporting or debunking the targets that were coming out of each other's groups, so we could actually come to a collective understanding of what here was reliable for each of these groups to go after, as well as the potentially the private funders as well. So we did this in 3 ways. The first was, of course, open data sharing. Data was shared early. It was meant to come into the system within 3 months of coming off of the data generation machines and become immediately available both to the consortium and to external investigators as well to use. The second thing was to thoroughly validate the bioinformatic tools that were being used as inputs to make the inputs that went into the modeling initiatives that were going on. And then the third was this concept of evaluating consistency in the scientific claims, in this case, both the network models and the targets coming out of the program. So I want to start by talking a little bit about what we did with open data sharing. So data, of course, needs to be shared in a way that is under fair principles, so it can not only be looked at, but actually effectively reused. And so as we thought about how to share data, we ran into many of the same issues and used many of the same solutions I'm sure lots of you use to make sure that the data was effectively reusable for others. And I just want to give you a sort of example of the kind of data I'm talking about here. There's lots of different data sets being shared through this program, but the ones that I think that are the flagships of this project are these 3 data sets. So the ROSMAP data set, the Mount Sinai Brain Bank data, and the Mayo Brain Bank data. And each of these is human brain samples, often across multiple regions, for which at this point, multiple layers of omic data have been collected. So we can start to use this data to do modeling of molecular state. These studies vary a little bit in terms of sample ascertainment. So we do have a collection of samples coming both from individuals diagnosed with disease, individuals without disease, and individuals with adjacent diseases as well. So we can start to compare across those various groups. And so as we've moved forward, we built all of our data sharing processes based on those 3 data sets, and then the adjacent ones being generated by those same groups. But since then, actually, this entire data sharing initiative has expanded, as Susanna mentioned, to include data coming from a whole suite of other consortia focused on omics data that are funded by the NIA. And so at this point, I think we're serving 84 different grants who are using this approach, this platform for data sharing. So what does that mean then? It means that our data sharing infrastructure needs to support a whole diversity of different data types, because this is not a program where we're working together to build one single collective data set. In fact, what this is a whole set of individuals building their own data sets with many diverse specimen types and data types and all sorts of different levels of processing. And so at this point, we have data from over 60 assays and in 50 different file formats. And so we have to find a system that's flexible enough to enable and support all of these individual projects, but in a manner that allows the data to actually be easily discoverable, searchable, and reusable. And so we've built an infrastructure which is based on our own in-house data cataloging and storage system called Synapse. Synapse has been under continuous development since, I think, 2012, and we use it across a whole host of different data sharing projects. And as we worked into this project, we started to build a whole series of applications on top of Synapse. Again, I think this probably looks pretty familiar to you because it's very similar to how data management and sharing is done across many projects. So we've built a web tool called the AD Knowledge Portal that enables search and discovery for the data. We have a program that allows the consortium members to work together for data processing and analysis in the cloud. We have a results explorer. So this is meant not just for the bioinformaticians to find the data, but actually for biologists and clinicians to be able to understand and interpret the results. And we had to put a lot of work into how we effectively scale in terms of data ingestion and validation down at the bottom. So at this point, the AD Knowledge Portal is enabling exploration of data coming from 7 NIA programs, and you can see some of the statistics here in terms of what that actually means. We have a growing number of portal users. This data is actually fairly specialized in terms of the kinds of questions you might want to ask about it, and so we're pretty pleased to see that we robustly have sizable data use. You can see that in terms of the number of active users. We have consistently about 1,000 users, and that's been true. That's been true over time. They're not always the same 1,000 users, but consistently that's what it's been looking like. They come from all over the world. This graph doesn't show it, but we also find that there's a pretty sizable group of individuals using this data coming both from the academic sector as well as from the private sector as well. And we've been doing this long enough that we're able to begin to see data reuse in the terms of publications coming out of use of the data. And so there are a whole suite of things here, which are uses of the data that we wouldn't have thought of ourselves, the investigators in AMP-AD wouldn't have thought of themselves, but are being performed using the data. And as Susanna mentioned, she's starting to leverage the data to start whole other programs where analysts can use the data to do other things, such as the drug repurposing study that she mentioned, program that she mentioned. For those that are interested, I wanted to give a high-level diagram of the functions of the Synapse infrastructure stack. And really what I wanted to note was this is a system that's used not just for this suite of NIA programs, but actually we use it across a whole suite of other programs at the NIH. And so I exemplify some of them here. And what this means is the data and the tools are all located side by side and annotated in similar ways, so we can easily start to do some comparative analyses across consortium and even disease states as well. One of the ways that this has been best leveraged within the programs at the NIA is actually being able to combine this data, this brain data coming from the NIA funded programs with the data coming out of the NIMH funded programs, also from brain coming from Common Mind and PsycEncode. And so we're able to start to do brain level analyses really at scale because all of this data is accessible within the system. Okay, so that's it for data sharing and reuse, or data sharing, right? The data is open, it's being used within the consortium, it's being used outside of the consortium, it's available. And the goal for that was both to enable transparency and independent evaluation of the results coming out of the consortium itself, as well as, of course, to catalyze secondary analysis. So what I'd like to turn to in the second part of the talk is how we, within the consortium, have used the data coming across all of these different sources to do community-based analysis. And I said in the beginning, we did this in 2 different ways. And so the first thing we did was to start to bring together these data sets to both benchmark the methods that were being used and to create reproducible and well-powered outcomes for those analyses that are typically the inputs for the kinds of network methodologies that the consortium is using. And so these are simple things, simple analyses like differential gene expression or EQTL analysis. As I said, having access to brain samples from all of these different consortium in one place has allowed us to do an EQTL analysis at a scale that was never possible before. And so doing this in a harmonized and robust way and providing those results, both for use by researchers within the consortium, but also, of course, for researchers outside of the consortium, is one of the immediate ways that we're able to provide value. It's pretty clear there are standard methods for many of the things I'm showing here, and so it's not necessary for everybody in the field to redo these over and over again. We can do them once in a transparent and clean way and provide that for others to use as value added. So then the second thing we do, which I'll spend some more time talking about, is the following. As I said, the problem statement when we got to the third or fourth year of this consortium was all of these different groups were using amazing methods that they had developed in-house to develop network models and make target predictions. And it was not possible, because they were all using different data and different methods, to really evaluate their results independently. And so what we came together to do was a meta-analysis where we ran all of the algorithms, which you see here, against all of the data sets that we had access to, so we could evaluate the consistency of the network models that they were building independent of algorithm and data source. So what does this look like? So it looks like this. What we found was the AMP-AD program was identifying approximately 5 biological domains that were impacted in human brain by Alzheimer's disease in a manner that was consistent across all of the data sets and all of the methods that were being used. And we could take this actually fairly simple meta-analysis approach and start to map that to what the individual groups were doing. So the first thing we found was by comparing this set of biological processes with that which has been previously reported in terms of gene sets related to Alzheimer's disease, we could see that the top 3 of these biological domains were highly represented in the literature and had been well studied before, right? Back to our first point about wanting to expand beyond things that are well studied. We have 2 biological areas here that actually are not all that well studied outside in the literature. And so what you can see actually is that the AMP-AD groups themselves, although they picked individual targets to go after, they all consistently picked targets coming out of these new domains, right? So they are consistently seeing the same understudied biology, and they are pointing their experimentalists at that work to go after. And I can tell you that one of these domains is focused on oligodendrocytes and myelination, and the other is focused on intracellular processes related to splicing and protein degradation. So why is it that there are these 2 areas that are showing up in this data analysis that weren't seen elsewhere? And in particular, in genetic analysis. And one of the reasons we think may be the following. As we started to analyze these various domains to look at what their characteristics were, we identified that these 2 new domains were actually inconsistently altered in Alzheimer's disease in female samples versus male samples. Versus if you look at the top 3 domains, actually there is quite some consistency there. And so you can imagine that a meta-analysis or any sort of analysis that didn't take sex into effect may in fact not identify these bottom domains. So if you put all the samples together, you will still consistently see the outputs in the top 3, but not the bottom 2. And so we were able to start to start to tease this apart. And this was highly valuable to all the research groups as they were starting their own work within these two domains. What else can we do with this information? Well, all of these groups, as I said, were supposed to be doing experimental validation, and they were doing this in a whole series of different systems. So certainly iPSC lines, fly models, but a lot of people were starting to work in experimental mouse models. And so we can now take these 5 domains, and actually we broke them down into a couple of dozen subdomains that are consistently seen in the human brain as a signature of Alzheimer's disease, and we can start to evaluate them across experimental mouse models, including those that have been used consistently and are seen in the literature all the time, and also the new ones that are being built through the NIA's mouse modeling resource Model AD. And so why is this important? Well, we know that the mouse is not a great surrogate for Alzheimer's disease, but if what you're trying to do is understand biology around a molecular domain, molecular mechanism related to Alzheimer's disease, you don't need the mouse to recapitulate all of the characteristics of Alzheimer's disease. Actually, what you need the mouse to do is to recapitulate that actual molecular mechanism so that you can study it in detail. And so we're able to use this this landscape map of what molecular mechanisms look like in the human brain and map it onto the mice so that our researchers within the consortium and researchers outside of the consortium as well can use this information to pick the right mouse to do the experiment that they're wanting to do. Another thing that we've been able to do now that we have this harmonized data and this characterization of the human brain is to start to use it to try to tease apart how Alzheimer's disease might be changing brain across disease stages, going from a control or on diseased setting all the way to late stage disease. And so what I'm showing you here is a pseudotime analysis that was performed on the data using manifold learning that was able to start to tease apart where certain molecular dysregulation might be happening in this stage of disease, so we can start to talk about what molecular pathway you might actually want to target at what stage of disease. So the last thing I want to talk about today is what do we do with all of this information? We have talked about a whole series of really tantalizing bioinformatic hypotheses that have come out of this work. And we are doing our best to use open science approaches to verify those hypotheses and to provide the evidence that lets other bioinformaticians independently evaluate these hypotheses as well. But ultimately, what you really want is for an experimentalist to pick up one of these hypotheses and start to study it using their set of tools to move this down the line. Again, back to this first slide one of the first slides that I showed, we don't want to just identify new targets or genes. We actually want to catalyze the whole community to start to study these. And certainly we can just pick them up and start to study them themselves. But in order to build the amount of evidence one needs to really get a target effectively over the hump of validation and into a drug campaign, you really need information coming from multiple lines of evidence across multiple laboratories in the community. And so you need to catalyze that. You need to inspire that. So how are we doing this? Well, first, as I noted before, we have this open platform that we use to openly disseminate the targets coming out of these groups. And it doesn't just list the targets, it provides all of the evidence that was used in their target nomination. It also provides a whole consistent set of evidence coming out of the various bioinformatic analyses that have been done. And increasingly, we're adding in experimental analyses as well. And so we're trying to create a community hub where we can start to objectively aggregate and evaluate the evidence around various targets so that there's a place people can go to look at this and think it through. This is, I think, really important, but it's not sufficient to get experimentalists to start to work on these targets. And so our other hypothesis was that one of the things that very much limits an experimentalist in moving into a brand new domain is that often understudied areas or understudied proteins simply don't have available access to well-validated reagents. And so our hypothesis here is that if we make well-validated, high-quality experimental reagents and put them completely in the open domain for use, that this will itself catalyze research into those domains. And this has been well proven by the Structural Genomics Consortium, who has been taking this approach for more than 10 years. And their work, which started in structural biology and has moved into medicinal chemistry and beyond, has been used to catalyze thousands and thousands of publications, many clinical trials, and actually several drug campaigns. And so this is we partnered with the Structural Genomics Consortium, and this is an approach that we took. So this is the inception of the second project that Susanna mentioned I'm involved with, a Treat AD Center. This is a group of bioinformaticians, structural biologists, medicinal chemists, biological assay experts who are coming together to develop high quality enabling research tools and drug-like compounds, so small molecules, to put in the open domain explicitly around some of the target hypotheses coming out of AMP-AD with the goal of catalyzing research in those areas. Our process looks something like this. We take the AMP-AD targets, we aggregate that with a whole series of other data resources that we have access to do therapeutic hypothesis selection. We're looking to select hypotheses for which there is reasonable evidence from the data analyses that we're doing, but very little literature on. So effectively a dark target. Now, I could talk for a whole hour about how you effectively select dark targets when you're explicitly looking for something that is understudied. That also means you don't have the kind of evidence you would necessarily usually use in target selection. And so that's been a very active area of research for us. Regardless, given the selection of a set of therapeutic hypotheses, right, we nominate targets within that hypothesis that we think that if we were to perturb, they would allow us to objectively evaluate the hypothesis, effectively de-risk this understudied hypothesis, not necessarily with the goal of running right down the line into a drug discovery campaign, but actually with the goal of providing the evidence the community needs to know whether this is a reasonable hypothesis to go after in more detail or not. From that, we build a set of reagents, which we call a target enabling package. And then for a smaller set of these, we are actually building chemical or biological probes that will enable in vivo investigation. And again, I said, all of this gets openly distributed without IP. So what's a target enabling package? Well, we've found that includes a whole series of experimental reagents, antibodies, purified protein, crystal structures, knockout and knock-in cell lines, et cetera, assay protocols, et cetera, all the way down to the chemical probe. And I'm not going to talk in detail about any of the work that we've done. The experimental work that we've done, I think that's probably out of scope for this group. But suffice to say, despite the challenges of COVID, we're about 2 years into this project, and we are starting to see reagents make it into the open domain. So a question that we get frequently is this. We are effectively building target validation and early drug discovery campaigns around bioinformatically identified hypotheses for which there is not yet sufficient data. So none of these hypotheses are at the point where in the traditional drug pipeline, they would actually get picked. There's not sufficient evidence. And so are we actually putting the cart before the horse? Why would you build chemical probes for a protein on which we actually don't know if it's useful in Alzheimer's disease treatment or not. And actually what we've found, what the Structural Genomics Consortium has found over the years is the act of having these high quality tools, including small molecule inhibitors, is essential to be able to effectively explore therapeutic hypotheses in a realistic manner. And so we are building these reagents that are traditionally thought to be really for drug discovery, but we're actually doing it to support target validation itself. I was going to talk a little bit about how we prioritize dark targets. So this would be back to reuse of the data coming out of AMP-AD and all of these other resources. I think in the interest of time, I'm not going to do that. I think I'll simply say that we have built out a bioinformatic pipeline through which we identify mechanistic hypotheses, and then we overlay the bioinformatic evidence we're getting both from our own analyses as well as from all the AMP-AD analyses on top of that, so that we can be very effectively picking so we can effectively be picking a potential new target mechanism as a whole, and then, as I said, pick multiple targets within it that help us perturb that mechanism and understand it. These are the sources of our first 4 set of hypotheses, and in green you can see the targets that we are working on building reagents for right now. Put this out there because these reagents are going to be coming into the open domain. If there's anything here that's interesting to you or your colleagues, we would love to hear from you to make sure that we're building the reagents that you think you need and to get your input on how we might do the work that we're doing. Our strategy is to build what we call a basic TEP, which is sort of the essentials of what you might need to do some basic cellular analyses for lots of targets. So 20 to 30 a year. And then the full TEP, which is much more expensive and time consuming to build, includes a small molecule inhibitor. We will do for a smaller set of targets. And so again, here's the list of things we're working on. So I'll conclude there. I think what I hope that I was able to convey to you today is that there are opportunities to use open science, data sharing, data reuse, and I think also importantly, the intersection of data-intensive scientists with experimental or clinical scientists to start to get at some of these gaps we have here. I'm talking about in the field of AD translational research, and we have been, I think, I hope I've convinced you, successfully using many of these techniques to specifically address the questions that the NIA is seeking to address within their translational research program. I'll end by thanking first my team at Sage Bionetworks, represented here, as well as the full TREAT-AD team. This is a tour de force of amazing scientists internationally across many different programs. Yeah, thank you for your time. I appreciate being able to talk to you, and I'll end there.
Thank you so much. Laura, this is an amazing amount of work that the team has done to create data science infrastructure and material science infrastructure and resources for this community. There are 2 questions in the Slido that I'll ask out. How does this approach affect the commercialization potential for any of these targets?
Yes, that's a great question. It's our expectation that the reagents we are creating are not themselves what will be the drugs in the future. They are reagents and inhibitors that will be used to query biology and probably any drug that comes out of that. And we think all of that information needs to be in the open domain, and anything that comes out of that, a drug may be built on top of that evidence. Having said that, the Structural Genomics Consortium is also initiating a new campaign to do open drug discovery, and they have a whole sort of legal framework around that for how they think that actually won't impact commercialization. So if you're interested more in that, look up Al Edwards in the Structural Genomics Consortium. He's running an amazing experiment in terms of whether and how it works to build drugs in the open domain that are still that can still get to market.
Thank you. Good question. How much engagement have you gotten from the basic research community in your portal?
Yeah. So it's a little bit harder to gauge because that information is openly available on the web. We do find that and so we can't track users as well. We do find that we do get not on the order of thousands, but on the order of hundreds of people using or accessing the data that's out there in any given week. I think that maybe I could answer the question in the following way as well. We very frequently run booths at conferences that have basic neuroscientists working at them, and so we're able to talk one-on-one with neuroscientists and individuals in the field of Alzheimer's disease to show them the information and get their feedback, and we find that they're incredibly receptive to this approach, and that they find the data to be useful for them in their processes. So how do you aggregate that community is maybe a separate question than what you're asking, and one that we're actively working on. And I would love any input anyone else has here on that. But we do find the data is or the results are very useful. And we do hear sort of anecdotally about figures from the portal being used in grant applications, which is like exactly what we want to see, right? This is helping people support the hypotheses that they want to go after.
Wonderful. For some of us, this is the first time that we've seen an open reagent sharing capability. So do you see a lot of use of the reagents that you're providing?
Yeah, so we're so early in the project that the reagents really haven't even hit the market yet, the market. So I can't answer that question yet for the reagents we're creating, but I can use the previous evidence of what the Structural Genomics Consortium has seen with the reagents that they've created. They make them incredibly easy to use, so you won't have to come to us to get the reagents. We're going to have them available through the common vendors that you would use, and they do see them taken up. And as I said, they see thousands of publications using their reagents and multiple drug discovery campaigns that have come out of them. So there is quite a bit of evidence that suggests that this will be effective.
Fantastic.
Susan, if I may add to that, actually,
Yeah,
while
please.
the TreatAD reagents have not hit the proverbial street, the sister centers, model AD centers, which are developing animal models as critical reagents for research, by design, they're also open science, open source, and all the animal models are pushed out, available. There are about 50 now, and can be accessed just with a shipping fee from JAX Labs. No IP barriers to access, licensing fees, etc. And they're taken up by academic labs and industry, including pharma. So that we know.
Thank you, Susanna and Laura. Another question that just came in, how much new methods and data types would you be integrating into the portal?
Well, so in terms of data, we integrate whatever data is coming out of any of the groups that are involved in these consortia. And so I think it's, it's dozens of different data types. In terms of the analyses that we do in terms of analytical coordination, we do specifically focus them where we see, on a more limited set of the data types. And the harmonization efforts of data coming in, we've really focused on some of the common omics analyses, RNA-seq, proteomic, the DNA sequencing, some of the epigenomic assays, because those are the assays that are most consistently coming into the portal. So there's a two-tiered answer to that question. We take all kinds of data. In terms of data that we specifically harmonize, it's a smaller subset of the more common data types.
Thank you. There's a good question here about the news. Okay, I might actually have a little bit of help here understanding the question.
Yeah. So regarding aducanumab and data sharing. Yeah. Right. So that it's an interesting so I interpret this question to say, is there any ability to access data, which right now would be entirely data that Biogen has, to uncover subtypes of individuals who might actually benefit from this, either clinically or in terms of amyloid? That's a great question. I'm so interested to see what the data looks like. And here I'm really interested in like electronic health records over the next couple of years as we look at individuals who are being treated with this. I think this is a great thing to do. It's a little bit out of my my domain. Susanna, do you know anything about sharing of the Biogen data?
Not of the Biogen data, but we have a big push and have embedded requirements for sharing data from our NIH-supported clinical trials, both early stage and pivotal trials, both placebo and treatment arms. And for pivotal trials, we insist on sharing screening data, which is prior to the ending of the trial. And so any trial that has any amount of NIH funding, could be 1%, 10%, whatever, they have to agree to those terms. It's been hard to break that barrier and get access to data and biosamples from legacy trials from industry, including failed trials. And that is a tremendous untapped opportunity of what we can learn. And we try to incentivize that through some funding opportunities where we encourage academic labs and pharma to partner, but so far, no takers on that.
You mentioned something that is intriguing, Laura, which is the integration of electronic healthcare record, and maybe even real world type data with the omics data that you are collecting now. Have you thought, or are you planning to do that, or is that just right now not within the possibility?
Yeah, I think that's so interesting, and absolutely where we need to go. I think for the specific data we're talking about here, there are small subsets of it that come from some of the that come from individuals for which there is other data accessible, and in particular through the AD research centers, which are supported by the NIA. And there are some conversations going on about how we might be able to integrate data across those various domains for those subset. But a lot of these human brain samples come from brain banks for which there isn't other information. Yeah.
Thank you. Well, I want to thank you and Susanna for coming today to our ongoing data sharing and data reuse seminar. Again, as a reminder, our next seminar is July 9th, Dr. Mark Meeuwsen, to talk about tools for making data more fair. We will certainly be posting your seminar fairly soon, and I hope that you'll come back and tell us in the future about some of the successes of the data sharing and data reuse that you're doing in the AMP AP program and the reagent resources that you're providing, which is all pretty cool.
Thanks so much. I really appreciated being here, and I'm glad to know about this seminar series. I think I'll start to attend.
Yeah, please do. All are welcome.
Thank you, Susan. Thank you, Laura and Aaron.
Thank you. Thank you, everyone. Have a wonderful weekend.
You too.